So, do you remember a few days ago when I Googled metabolic pathway map poster and I showed you a PDF and it was enormous? If we printed it out so that it would be legible in like minimum 18-point font, it probably would take up all of the front of this room. We had to zoom in, and even then it was pretty complicated to observe. One of the things that if we had spent some time looking around that, and maybe I'll do this next time, we would have seen the citric acid cycle. I think there was a circle that we basically identified as the citric acid cycle. And had we looked at that long enough, we would have been able to find glycolysis, but there was a whole lot other stuff going on. And the point I want to make is that metabolism is all interrelated. We've looked at various pathways as though they are just linear, following glucose down to pyruvate, to acetyl-CoA, to the citric acid cycle. Within the citric acid cycle, you're going to see that there are various metabolic pathways that generate some of the intermediates in the citric acid cycle, creating a web of related reactions. One thing we're not going to talk about very much is you can actually go backwards from pyruvate up to glucose. Your body needs to do that when your blood sugar runs low and you're not eating carbohydrates to feed your brain. So your body synthesizes glucose, sends it out into the blood, and sends it to your brain. So it's this vast network of interconnected pathways. We can't hope to talk about all of them. We want to talk about enough of them so that you have an idea of what happens to triglycerides. So check, we've done that. Carbohydrates, check, we've done that. And today is what happens to amino acids. Yesterday we talked about what happens to peptides and proteins in your stomach. We talked about the serine proteases with that catalytic triad that can hydrolyze amide bonds. That wasn't a new mechanism because it's essentially the same mechanism that you saw when we used a serine lipase to hydrolyze the esters in a triglyceride. So today we'll talk about what happens once the amino acids in a peptide or protein are all separated from each other. You've got your regular amino acid. How does your body metabolize that? And that's going to involve what's called transaminase chemistry. That's the old term for it. Alternatively, you could call it aminotransferase chemistry. And all of this is also closely related to how your body handles ammonia. So transaminase chemistry is basically moving amino groups around. And you're going to see specifically that what we're basically doing is moving an equivalent of NH3. NH3 is ammonia and it is acutely toxic. So you can't just put ammonia in your blood and hope for the best. There's some problems that would occur if you did that. And so your body has a system for transporting ammonia and then also for getting rid of it. The system for getting rid of it is called the urea cycle. It's another metabolic pathway. We're not going to talk about it, but it converts ammonia into a molecule called urea, which if the word sounds like urine, and that's why, because it's excreted in urine. But what we'll talk about today, you will see, involves how your body transports basically an equivalent of ammonia around without it actually being ammonia. At the same time, this will also explain what your body does with amino acids. Before we get there, I need to remind you of a couple of things you've learned. So some of these things are shown on your quiz, and I've got the figures here. A couple of weeks ago, we talked, and you may not remember it because it happened pretty briefly, 2 different times, about the reaction of an amine or an amino group with an aldehyde or a ketone to give a functional group called the imine, I with a I-M-I-N-E, imine. Somebody wants to write a children's book with the title I is for imine. I think it would sell at least 1 copy, and probably exactly 1 copy, and I would buy it. So somebody out there make it happen. But An imine is what you get when you do a condensation reaction between an aldehyde and an amino group. So one example that you saw was when we talked about vision, and when we first introduced our mechanism handout, this reaction was in it. This is 11-cis retinal. This is derived from vitamin A, by the way. Vitamin A is the alcohol version, so this is the oxidized version of vitamin A. It has an aldehyde. There's your carbonyl carbon, there's your carbonyl oxygen, the LUMOs, the carbon oxygen pi star, and then the protein in your rod and cone cells that can absorb light. Before it has retinal linked to it, it's called opsin, and there is a lysine side chain. This happens to be the beta carbon, gamma carbon, delta carbon, epsilon carbon, and there's the auto deselect eraser. Ooh, that's useful. Let's see how well it works. So I can erase something. Oh, I like that. I've kind of wanted them to, that's actually more effective than what I wanted them to do. I wanted them to make the butt end of the Apple Pencil the eraser, so you can just turn it over and erase. But that works, just automatic de-erase works good too. Okay, sorry, I got derailed. In any case, this is the epsilon amino group from the protein opsin. It's currently in its ammonium form. It's gonna need to become the neutral amino form. Probably could get deprotonated by an enzyme in order to become nucleophilic, or deprotonated by solvent, whatever, by water to become nucleophilic. This is the nucleophile. I'm not really worried about whether you know the mechanism for imine formation or not. I think it's in your mechanism handout, but it's complicated enough that I'm not going to ask you questions about the mechanism of imine formation. It's enough now to understand what happens. The nitrogen attacks the carbonyl carbon. You get the carbonyl oxygen gets protonated, and ultimately you end up with an intermediate that looks like this. I'm just going to show you the intermediate so that what follows will make some kind of sense. Oops. If you don't erase effectively, you lose your chance. Well, in any case, here is what, I'm skipping ahead a few steps. Here is what the intermediate looks like. This is the carbon that used to be the carbonyl carbon. This used to be the carbonyl oxygen. This is your nitrogen nucleophile. If the OH group gets protonated, that turns it into a good leaving group, and the nitrogen lone pairs can kick off water as a leaving group. And ultimately, you end up, after a proton transfer, with this molecule, which is called an imine. This is the imine functional group. This is the nitrogen that was the nucleophile. This is the carbonyl carbon. Where did the carbonyl oxygen go? It left as water. So this is imine formation. I don't care that you know the mechanism. I do want you to be able to predict the products of imine formation. The starting materials for imine formation are either aldehyde or ketone, and then amine. And they react together to form an imine. Are there questions about what an imine is? Imines look like carbonyls, only the oxygen's a nitrogen, and the nitrogen's attached to one other group. And in the case of vision, attachment of the retinal to the lysine side chain via an imine allows later on, when this molecule absorbs light and isomerizes to the transform and the molecule moves, that allows that motion to affect the protein. Yeah? The two other hydrogens from H3O. So let's see, the nitrogen used to have 3. Now it has 0. So the hydrogens come from the amino group. Used to be NH3, now it's just N. Yeah.
They just transfer.
Yeah, I mean that's a mechanism question, but we can talk about it briefly, though I'm not going to ask you a question about this. So I'll try to just be quick about it. One of the things you could imagine doing is some base removes the proton, or some conjugate base removes the proton from the amino group to make an acid. Then that acid protonates the carbonyl oxygen as the nitrogen's attacking, so that gets you an intermediate that looks like this. So then one of the protons from, I'll just highlight the protons in like green, one of the protons in green is now on the oxygen. Then another one gets picked up by that oxygen. That process that I just drew may be mediated by an acid and a base, but this is good enough. That turns the OH into an even better leaving group, which gets kicked off. That will generate an intermediate that looks like this. This is called an iminium ion. And then water can steal the last proton from that iminium ion, and that's where you get the H3O plus from. Okay. Good. So again, if you didn't follow the arrow pushing for the imine formation, that's okay. I'm not going to ask you questions about it. It's actually not that bad if you practice it for a while, but there's just too many details, and you've learned a lot of mechanisms already. Other questions about this? Go ahead.
So you said an imine is it's like the carbonyl, but the nitrogen is in the oxygen spot.
Yep.
And it's bound to a carbon, but it's also bound to something else.
That's right. So in an imine, the nitrogen is doubly bound to the carbonyl carbon, and the nitrogen has another group here. And there's a lone pair on the nitrogen, so it has a 0 formal charge. There's tons more we could say about imines, but as long as you know how to draw them from the aldehyde and the corresponding amine, you should be good to go. In some ways, this is a little bit like Lego chemistry, where you take the blue and the pink atoms, the carbonyl carbon and the carbonyl oxygen, you separate them. The carbonyl oxygen is going to become water. You take the nitrogen, you pull off its protons, you put the nitrogen on the carbonyl carbon, click, that's your imine. You take the other protons, you put them on the oxygen, click, that's H3O+. All right? You can see this again in what we talked about when we did carbohydrates. We talked about blood sugar. We talked about the Maillard reaction for browning of various baked goods and cookies. Then we demonstrated that in class with real live cookies. But this is what happens to your red blood cells when your blood sugar is too high. Glucose is an aldehyde, and the glucose aldehyde can react with the N-terminal amino group of hemoglobin. This happens to be the N-terminal valine of hemoglobin. Here's the nitrogen that's going to act as a nucleophile. There's the protons that are on the nitrogen. Again, look at imine formation. Separate the oxygen, carbonyl oxygen, and the carbonyl carbon. Click. Then separate the nitrogen of the ammonium group and its protons. Click. Then put the nitrogen in the place of the carbonyl oxygen, and then put the protons on the oxygen. And you've got yourself an imine and H3O+. All right? So that's imine formation. Yeah?
It would be helpful for me if you could sit over a timer, like make a list.
Oh, okay. Let's make a list. Good. So conceptually, this isn't, please understand this list is not a mechanism. If you want the mechanism, I can draw it for you. But I'm not going to ask you questions about it. Conceptually, you're going to break the carbon, oxygen, sigma, and pi bonds. That's click 1. Then click 2, remove protons from nitrogen. Click 3, oh, this, I guess that's unclick. Click 3 is make nitrogen, oxygen, sigma, and pi bonds. And then, okay, suddenly I'm getting, if it was unclick 1 and 2, then it should be click 1 and click 2, whatever. And then lastly, you're going to make the 3 oxygen, hydrogen, sigma bonds. This will give you the imine, this will give you H3O+. All right, so now let's practice. And this will get us to the first step of amino acid metabolism. So if you're in a situation where you need to start metabolizing amino acids for energy rather than to synthesize other proteins, whether that's breaking them down into acetyl-CoA and going to the citric acid cycle, or alternatively using them as starting materials for glucose synthesis. Either way, the first step is imine formation. So let me introduce you to a new aldehyde. I don't think you've seen this one before. Maybe you have. I don't think it's come up. This is called pyridoxal phosphate. And it is an important participant. I guess you could call it a cofactor for for transaminase reactions, and it functions via imine formation. The abbreviation is PLP. You get pyridoxal phosphate in your diet as vitamin B6. So the B vitamins are really important. We've talked about almost all of them. The ones we haven't talked about, mostly because I don't understand them, are vitamins B9 and B12. But we've done B1, 2, 3, 5, and 6. 7, 9, and 12 we haven't done. I don't know what vitamin B4 is. 4 must have just disappeared or got called something else. In any case, a lot of the B vitamins are heavily involved in metabolism, and you've seen a lot of them so far. There's actually a question about that on your problem set to identify some of these key cofactors as having components of vitamins. And I've tried to point those out as we've gone through the notes, and I can mention them again sometime as we move on. But the key functional group for right now on PLP is the carbonyl carbon and the carbonyl oxygen. Another thing to pay attention to later is this ring. It's not a benzene ring because there's a nitrogen in it. It's called a pyridinium ring because it's positively charged. The nitrogen's protonated, has a positive formal charge. You've seen something like that before in NAD+. And in a way, you will see later on today, not right now, that this ring is important. It's an important place that electrons can go. It's a potential, what is sometimes called electron sink, meaning a place that can stabilize electrons. For right now, though, focus on the aldehyde. And in your drawings of imines, for right now, it's totally fine to draw the imine, to draw all of this stuff over here as an R group, okay? So feel free to, what I want you to do now is draw the imines that you would get from each of these amino acids. We'll start with alanine, valine, and aspartate. Basically, once you've done one, you've done them all, but we're going to learn about the transaminase reactions of each of these amino acids, so it's useful to get started on them. Okay, so take a few minutes, and based on what we've talked about, draw the imines. You can abbreviate PLP as an R group. You'll need to draw the carbon that's the aldehyde carbon and the carbon you'll need to at least have the blue carbon involved before you abbreviate everything as R. All right, go ahead and get started, and then I'll come around and answer questions if you get stuck. I'm standing here, not even dead. Go ahead. Okay. So where's the Yeah, there's a hydrogen implied. Yeah, folks, that aldehyde that I've drawn, there's a hydrogen that's implied but not shown. Feel free to draw it in if it helps you recognize that's the aldehyde. Yep. And then your amino group is that one. Yeah, pretty much. So what happens? The things that happen. I mean, there's no first, second, or third. We're just going straight to the end of the draw. I don't have to draw like this or anything. Exception is you got to break the carbon partition. That's my one. So from like the carbon, the aldehyde, put the carbon in the oxygen. Get rid of the hydrogens on the nitrogen. Put the nitrogen where the oxygen was. And then take all three hydrogens. Put them on the nitrogen. Nitrogen goes in place with the carbon oxygen. Yeah, you bet. Others? Perfect. Yeah, great. That's right. Oh, yeah, sorry folks, I didn't want to draw PLP up here like 3 times. So these are 3 separate reactions. It's the reaction of alanine with PLP, and then the reaction of valine with PLP, and the reaction of aspartate with PLP. I just drew the structure of PLP. Oh. Yeah, sorry. Okay, other questions or worries at this point? I mean, questions or worries related to immunes, I guess we would start there. We can talk about other questions and worries if you want. Yeah?
We were to just shorthand this, right? Top R
Yeah, the question is if you want to shorthand things by doing an R group for the side chain and an R group for on PLP, wouldn't it be the same thing for all of them? Yes. That is what I wanted to show you. Generalizing the structures, yeah. So on the first one, I'll just draw the full structure for you without the R group so you can have an idea of what this looks like. The ring portion doesn't change in imine formation. It's only the aldehyde portion that changes. So you can draw the PLP portion exactly the same way each time. Again, I'm showing you how you would draw it if you were not going to abbreviate. Let's do alanine. So this is the carbonyl carbon of PLP, and it's now going to be doubly bonded to the nitrogen of alanine. That nitrogen is going to be neutral, no hydrogens. Here's the alanine side chain and the carboxylate group. If you want to, you can fill in the proton that's on the alpha carbon of alanine that was implied but not shown. Here is the nitrogen, and then of course you're going to make H3O plus here. Where did the carbonyl carbon go, carbonyl oxygen go? It went onto water. Where did the protons from the nitrogen go? They're over here on the oxygen. Okay, questions about that? So if you wanted to abbreviate for the other one, it would look like this. R here represents the rest of the PLP ring. That looks kind of like a flamingo, a gray flamingo. Sad. This is the carbonyl carbon, and it should be bonded to the nitrogen of our amino acid, which is here is valine. Here's the valine alpha carbon, the valine side chain, and the carboxylate. If you want, you can draw in the proton that's implied but not shown on both the aldehyde and the alpha carbon. So that's what the valine amine would look like. And then for the aspartate, it would look like this. Everything's the same. You just fill in the aspartate side chain. And I think what you noticed, Ange, was that all of these, if you let R prime equal the amino acid side chain, all of them follow this basic pattern of this basic structural pattern, right? They all follow that. So that's actually an important observation. With amino acids, nature can use similar chemistry to process different amino acids in basically the same way. It tolerates variation in the side chains. There will be different enzymes that do this for alanine versus valine versus aspartate, but the players will be very much the same. Yeah?
I just wanted to know if the net charge on all these would be negative 1.
The net charge on what? Well, let's see. PLP is neutral. A plus here and a minus here. The amino acid is neutral. So overall, I think you see that the net charge, considering both the imine and H3O plus, is still neutral. Am I missing something?
Does the N in the middle have a plus on it?
Oh, the N in the middle does not have a plus on it because it's got a lone pair. It owns 5 electrons. Nitrogen normally comes with 4, so 5 minus 4 is plus 1. Holy Toledo, Batman, that was every word I just said. Well, half the words I said were wrong. Formal charge on that nitrogen. That's so embarrassing. Formal charge on that nitrogen. It currently owns 5, And then how many valence electrons does nitrogen usually come with? 5. 5 minus 5 is 0. The net charge on that nitrogen is 0. So sorry. Okay, we good on imine formation? What other? Go ahead.
On the, like the list you gave us on click 1,
Yeah.
it's like make the N oxygen.
Oh,
It's
yes.
not directly bonded to the.
No. That was meant to be NC. I think that is now 2 mistakes in a row is now extra credit point for everybody that's here today. Yeah. You guys are getting kind of like lying in wait to catch me in something. But you know what? I suppose that's how you feel when you take my test. So what goes around comes around. All right, yes, you are right. I am so sorry. Let's review the rules for imine formation as correctly stated now. First, unclick the carbon-oxygen sigma and pi bond. Unclick the carbonyl. Second, unclick the nitrogen from its protons, the amino nitrogen from its protons. Then, now on clicks, click 1, make the nitrogen carbon sigma and pi bond. I'm so sorry. And then click 2, make the oxygen hydrogen sigma bonds. I think that's correct. Yes?
Can we just remember the mechanism if we don't want
If you don't want to remember the clicks and the unclicks because you don't trust me to say it right and you know the mechanism, you're good with it, yes. That is why I can't say the clicks correctly because I just invented it today, but I learned the mechanism 20 years ago. Yeah.
Are you recording?
Am I Oh, I see what you're doing. We want a double or nothing. Yes, I'm recording. All right. Very good. Other questions? Thank you for pointing that out. Other questions about imine formation? Okay, so that's step 1. This is catalyzed by an enzyme called aminotransferase for each of these, and there's a separate aminotransferase for many of the amino acids. For example, alanines is called alanine aminotransferase, unsurprisingly. And valines would be called valine aminotransferase, aspartates would be called aspartate aminotransferase, illustrating how enzymes are named again with the substrate first and then what it does, what the enzyme does next. I'm not actually sure what the valine enzyme is called, and I'm not going to ask you to say what, I'm not going to ask you to name the specific enzymes anyway, though I do want you to know that it's an aminotransferase that does this. By the way, that name should indicate that the amino group is going to go somewhere. It's not anywhere yet. It's just been tied up as an imine, but that's the first step. Okay. Let's now, I want you to take, I'll show you the next step of the mechanism. And the goal is to remove the amino group from the amino acid and take it somewhere else. So right now you don't know where we're going. I haven't shown you the final products of the reaction. We'll review that a little bit. What I want to do now is take the structure from the imine. We'll call this the alanine imine. And we'll copy it and put it on the next page. I'm going to show you something for alanine, then I want you to show it for at least one of the other amino acids. Your quiz says do it for all 3. Now that you know that it's the same for all 3, just the side chain's different, you only really need to do it once. Let's be just a little bit more specific about the alanine imine. The carbonyl carbon in this imine came from what kind of functional group? Was it an aldehyde or a ketone? It was an aldehyde, right? Because PLP is an aldehyde. So some people will call this an aldimine. And that's fine. I'm saying this because what we're going to do now is change the location of the imine. So, I want you to remember what you've learned a long time ago is that protons that are alpha, and by alpha I mean alpha to a carbon-oxygen pi bond or perhaps a carbon-nitrogen pi bond, are somewhat acidic. And the reason that they are somewhat acidic is their conjugate bases are resonance stabilized. That is terrible penmanship. So what I want us to do is use some base from the enzyme to deprotonate the alpha carbon, and we're going to push the electrons onto the alpha carbon, and then we're going to look and see how it's resonance stabilized. For this, you will need to draw the whole molecule. If you want to abbreviate something, then by all means abbreviate the side chain on the amino acid, but you're going to need the whole PLP. Okay, are you with me on that deprotonation? You see the lone pairs that are on the alpha carbon? Yeah.
Is this the tautomerization?
Yeah, this is the first step of the tautomerization on question 2. Notice that I've changed the line between the alanine alpha carbon and the side chain to just, instead of being a dash, it's now just a straight line. That's because this is no longer a stereocenter. The alpha carbon here will be trigonal planar sp2 hybridized so that the lone pair can be in a p orbital where it can overlap with the adjacent pi bonds. So notice that this lone pair on the alpha carbon is adjacent to multiple pi bonds. It's adjacent to the carbon-oxygen pi bond. It's adjacent to the carbon-nitrogen pi bond. And on the other side, you can see that the carbon-nitrogen pi bond is also adjacent to other pi bonds, which gives you an extended, what we would call an extended pi system of resonance or conjugation. We haven't talked a lot about that, and we don't have time to talk about it in a lot of detail. But as you know, resonance is stabilizing. Resonance delocalization is stabilizing. Let's just draw a couple of resonance structures from this intermediate. These aren't arrows in the reaction, but we're going to draw resonance structures that convert this intermediate into another resonance structure. One easy one would be to take the electrons on the alpha carbon, move them here between the alpha carbon and the carboxylate carbon to make a new pi bond, and then break the carbon-oxygen pi bond, and then electrons would go out onto the carbonyl oxygen. So that resonance structure, and that's one that we've seen before for other similar molecules, that would look like this, which tells us that the electrons on the alpha carbon are actually delocalized out onto the carbonyl oxygen. So that's a resonance structure. That tells me that the negative charge is not only on the alpha carbon, but it's also on the carbonyl oxygen. Now let's do the same, starting from, again, the same structure that we started with. We're just going to push arrows. And this time, we're going to go the other direction. I'm going to push these to form a new pi bond with that nitrogen. Yeah, go ahead.
For the structure you drew above
Yep, sorry about that.
And then there's still the possible
Yes. I feel like all of these mistakes are covered on the original mistake, but at some point I'll pass out more if I keep causing trouble. Yeah. On the old Sesame Street, there was a segment called Elmo's World. That may be before your time, I don't know, but Elmo used to talk to a strange guy called Mr. Noodle who was outside the window in his own sort of world. And Mr. Noodle's big bit was doing everything wrong so that Elmo and the kids could say, "No, not that way, Mr. Noodle." So I feel like his mistakes appeared to be deliberate. Mine are inadvertent, but if you'd like to say, "No, not that way, Mr. Noodle," I'll be embarrassed because I prefer that you call me Lord Vader. But you can insult me by, yeah. Anyway. So, with that all said, we can move the electrons to form a pi bond with the adjacent nitrogen. If we stop there, we'd have too many bonds to nitrogen, so we can move the electrons from this carbon-nitrogen pi bond elsewhere. I'm going to show you one possibility. A set of 3 additional arrows that pushes electrons onto the nitrogen of the ring. That nitrogen used to be positively charged. Once we put the electrons on it, it will now be neutral. And that's what I meant before when I told you the ring of PLP is an electron sink. So here is, a new resonance structure for PLP, and the point is just is that once you remove the alpha proton from the alanine aldimine, the resulting conjugate base is stabilized by resonance. Okay. Questions about that? We need to draw a third resonance structure. Ooh. Question withdrawn. No, I mean, that's one way. Which of these is a valid resonance structure? I've asked that kind of question before, so no, you didn't give me that idea. I already had it. Take a look at this starting structure where the lone pair is on the alpha carbon. Notice that we moved here to form a pi bond. We moved these over here, and these there to get this resonance structure, where in effect, the lone pair that used to be on the alpha carbon is now delocalized onto the nitrogen of the ring. Yeah. What if instead of pushing the electrons all this way, what if at this stage we had simply moved the electrons to what used to be our aldehyde carbon? Okay, so I'm going to just draw that resonance structure. We haven't drawn the arrows to get there. We're just taking them from the previous structure and stopping part way. Do you see how this is a valid resonant? Oh man, that eraser thing is kind of annoying. I thought it was good, but okay. So do you see how that's a valid resonance structure? that puts the lone pairs on what used to be our carbonyl carbon. If you summarize everything that we've learned from this process on one of the resonance structures, the original one that we started with, I'm going to erase the pink stuff. I thought I was going to erase the pink stuff. How do I need to disable that. The eraser thing is kind of annoying. All right, erase that, erase that. I want to disable that thing, but I don't know how now. Oh, okay, the double tap might help. Okay, in any case, I'm going to highlight in pink all of the places where resonance would show you that lone pair can be. Notice it's every other atom. That's just how resonance works. So what we're trying to communicate here is that the alanine aldimine can be deprotonated on the alpha carbon to give a resonance stabilized intermediate where pink indicates places where the lone pair / negative charge, depending on which atom it's on, can be. All right? With that in mind, let's show what would happen if we take this particular resonance structure where the lone pair / negative charge is on the other side of the nitrogen. And let's go ahead and protonate on that carbon. So that would look like this. Pick up a proton from the conjugate base of whatever, the conjugate acid of whatever base we were using to begin with. And let's look at what the product of that proton transfer reaction would be. And then just below, I'll summarize sort of what we did to get there. We started with the alanine aldimine. We got to an intermediate that looked like this. In which the alpha carbon was negatively charged. We recognized that by pushing arrows this way, we could put a negative charge on the other side of the nitrogen, of the imine. Here is the negative charge now. And now the alpha carbon of alanine is doubly bonded to the nitrogen. And at that point, we protonate on the negatively charged carbon, and that gets us to our product. Which is what I drew above, but I'll draw it again here just for completeness. Okay, questions about getting from the alanine aldimine to this product? It's really just two proton transfer reactions. And because it's two proton transfer reactions, we'll call this tautomerization. What we did with drawing all the resonance structures was to convince you that the presence of PLP makes it easier to remove the proton from that alpha carbon because the resulting conjugate base is stabilized by resonance. We then focused in on these two resonance structures of the same intermediate that show how the negative charge on the alpha carbon is also on the carbon that's adjacent on the other side of that nitrogen, resonance delocalization. And if we protonate on that nitrogen, we end up with a different product. Now, this product is still an imine, but notice that compare it to the starting aldimine, notice that the carbon to which the nitrogen is doubly bonded has changed. Do you see how it's on the other side? And notice that that alpha carbon is itself bonded to two other carbons. So we're going to call that a ketimine, different from ketamine, the drug, ketimine, or ketamine, because it's now on the other side. Or if you don't want to call it the ketamine, you can call it the other imine. Instead, you could call this one just the imine or the aldimine, and then the other one is called the ketamine or the other imine. Now, so far, it's just tautomerization or proton transfer. It may not make sense what we're doing, but it'll become clear in just a second as we talk about breaking apart that imine. Go ahead.
With the tautomerization, can you do it from any of the resonance structures?
You could, yes, you can push arrows successfully from any of the resonance structures to show the tautomerization. You have to, ultimately, it's this carbon here in pink that gets protonated, but you could show that from any of the resonance structures. For example, if I'm going to do this and then erase it just to make sure that the notes aren't disorganized, but I could have, instead of drawing this, I could have simply done this. And that would work too. Okay. In fact, since we're all about versatility, if you don't want to draw, I drew the intermediates because I think it's important for you to know why you can remove that alpha proton in the first place. But if you wanted to, you could draw the the deprotonation and the protonation steps all in the same step. I think some of you may like that better, and that's fine. To go straight to the ketamine, and that's okay too. Okay, other questions about that? All right, I think we probably need a break because that was a lot. I'm desperate to go on to the next thing because it's awesome, but let's just take 5 and then we'll pick it up again.